It's my esteemed pleasure to introduce Robin de Flor. We're all very happy he's here giving this talk. Robin did his PhD with Gael Chetelat in Caen, France, where, among other things, he was interested in hippocampal subfields and functional connectivity, both in the context of Alzheimer's disease, but also across the lifespan. Robin then went on to do a postdoc at UPenn with Paul Yuskevich and Dave Wolk, and also along with Laura Visser, who was there as well, and I guess the talk that he'll be doing today is a lot of the work that he did during that time, which a lot of which was based on looking at different pathologies in the hippocampus. And he is now back with Gael Chetelat doing a postdoc as well. So Robin, I think I covered your entire life there. Thanks for coming. And
so yeah, I'm going to talk about the medial temporal lobe, and more precisely, I'm going to talk about 3 different projects I've worked on during my 2-year postdoc at UPenn. So the first project is about the anatomy of the hippocampus. So we used ex vivo images to better describe the anatomy of subfields. And then I'll talk about a project about pathology and relationship between mixed pathology and medial temporal lobe atrophy in the context of Alzheimer's disease. And lastly, I'll talk about a project about brain networks and atrophy propagation, also in the context of AD. So just a little bit of background. I'm sure that you all know this, but the medial temporal lobe is made of different structures, including the amygdala, the hippocampus, the entorhinal cortex, the perirhinal cortex, and the parahippocampal cortex. And these regions are very important and have been studied a lot because they are involved in many cognitive processes such as episodic memory, semantic memory, spatial navigation, emotional processing. And they are also affected in many conditions such as Alzheimer's disease. So I'll talk a lot about this, but also aging and other neurodegenerative diseases, temporal lobe epilepsy, and depression. So a lot of people are interested in these structures. And the hippocampus is also made of substructures called hippocampal subfields, including the subiculum, the 4 fields of the coronary harmonies, so from CA1 to CA4, and the dentate gyrus. And interestingly, MRI sequences were designed with very high resolution so that we can see anatomical details and we can segment the different subfields, the different hippocampal subfields in vivo in humans. So here it's an example of a sequence and a protocol that have been made in the lab I'm working at the moment in France. So it was made 10 years ago by Renaud Lajoie, and you can see here that we are able to estimate the volume of the subiculum, CA1, and the rest of the subfields that were grouped together because we don't have enough details. But using this segmentation, we could, for example, compare the volume of these structures across different clinical groups. So here you can see in white LC controls, in gray MCI patients, which can be considered as a pre-dementia stage of AD, and AD patients in black. And we see that CA1 shows strong atrophy in MCI patient as compared to controls, which is also the case in patients with Alzheimer's, but there's no differences between these 2 patients group. The subiculum is also atrophied in MCI patients and tended to be more atrophied in AD patients, and the rest of the subfields were only atrophied in patients with AD. So this showed differential effects of AD on these subfields. So this is just an example, but many groups showed this as well. We also showed different effects of age on the subfields with a nonlinear relationship between age and the volume of CA1, which stopped decreasing at around 50, while the subiculum decreased linearly, and we didn't find the association with the rest of the subfields. So all of these things are just to illustrate that it's important to consider all these substructures, especially in the context of AD and neurodegenerative diseases. So that was the background. And now I'm going to talk about these specific studies. And I'll start with this study that was published last year in Hippocampus. So yeah, hippocampal subfields are defined based on cellular organization. So for example, the subiculum is made of widely spaced pyramidal cells, while the dentate gyrus is made of granule cells. However, protocols for segmentation performed on in vivo MRI rely on intensity features and geometrical rules, because of course we cannot see the cellular organization in these images. So we have to keep in mind that the subfields segmented in vivo are just an approximation of the real subfields that are based on cellular organization. And if you look at the literature, you can find more than 30 protocols that were defined in different groups. And these protocols showed a lot of differences regarding images. So for example, the resolution or the weighting, also the number of segmented regions, the location of subfield boundaries, especially the boundary between CA1 and subiculum is very different across protocols, and the field strength as well. Some protocols were defined for images acquired with 3 Tesla, while some other were designed for 7 Tesla. So for example, you can see here that this protocol defines 3 regions and is performed on proton density scans that are acquired with a 3 Tesla scanner, while the protocol by Laura Wisse includes 6 regions that are segmented on T2 scans acquired at 7 Tesla. So there are a lot of differences, and it's sometimes hard to really compare results from different groups because of these differences in protocols, and to replicate findings more precisely. But there is one common point, which is not necessarily good news. All these protocols are based on neuroanatomical literature, and unfortunately there is a mismatch between the orientation of histological sections, which are acquired following the ACPC axis, and most of in vivo MRI, which are acquired following the perpendicular axis of the hippocampus. So what you see in this literature does not directly translate to your images. Moreover, in these neuroanatomical papers, you only have one picture or sample, so it doesn't really take into account the anatomical variability. So in this project, we really wanted to take advantage of ex vivo MRI associated with histological sections to better describe the anatomy of the hippocampus and to be able to use this information for future protocols. So we used postmortem data that are publicly available and that were used in this paper by Wisse PNS. And it consisted of 9 specimens, so 3 patients with AD, 2 with other dementia, and 4 non-dementia control. And for each specimen, we had histological sections, and these sections were used. So first they were scanned, and then they were used in order to set boundaries to define the different subfields based on cellular organization. So this is the real real segmentation for subfields. And then the specimens were also scanned using ultra high resolution MRI that you can see here, and they were aligned with the histological sections, so that we could use the boundaries to perform the segmentation. That is the real segmentation based on all the histological and cells organization, you know. So it's the real real subfield segmentation, and we used this data to answer several questions that are not super clear and that are sometimes problematic in the literature between the different protocols. So I will not describe all these questions we try to answer, but I'm just going to talk about the tail a little bit and about the dark band. So first of all, we decided to focus on the tail because some protocol decided to not segment the tail. Some other do it. The anatomy is not consistent across this protocol, so there is a lot of mismatch between all these protocols, and we wanted to better understand the anatomy of this particular part of the hippocampus. So actually, we looked at the tail, so the posterior part of the hippocampus, and we could describe 2 types. So the first type is what we call body-like shape, because it really looks like the body of the hippocampus. And there is also another type, which is a bit weird, and it's not non-body-like shape. So this is also what we could observe in vivo. There is no consistency in the shape of the tail. But what is interesting is that when you look at the non-body-like shape tail, and if you just rotate it a little bit, you'll have a body-like shape. So it's the same hippocampus, but just with a rotation. So it means that the shape of the tail really depends on the rotation and the axis of the acquisition. So, and this is really hard to make sure, I mean, when you acquire your images, to make sure that you have this perfect angle to have this body-like shape versus not. So, for example, here we suggested not to segment the tail because of this problem. Then we try to describe the dark band. So the dark band is this band, which is often used, I mean, almost in all protocol, as a boundary between a CA1, CA, and the dentate gyrus. However, we don't really know what is the dark band. It could be SRLM, which is more part of considered as CA, or it could be the stratum moleculare of the dentate gyrus, which is here in blue. So some protocols include the dark band only in CA1. Other protocols split the dark band between CA1 and the dentate gyrus. So this is very problematic, and we really want to know what is exactly the dark band. And that's also very important because we know that CA1 is atrophied very early in AD, for example. So that's very important to be able to define it. So we used 3 different images for this. The MRI image, the histology image, and the segmentation, where you can see SRLM here in white blue, and the dentate gyrus molecular layer here. And then, so all these images were aligned and open at the same time in ITK snap. So we just draw a line from this point where the dentate gyrus meets the subiculum to the most lateral point of the hippocampus. This way to make sure that we did it exactly the same way across subjects. And we reported the line for the dark band here. We draw it, and it was automatically reported on the other images. So we could measure the thickness following this line of the dark band, but also the thickness of SRLM and TGSM. And we also could directly see where was located the dark band, because it's directly reported on the segmentation performed on histology. And what we saw was that the dark band was only included in SRLM, never in the dentate gyrus SM. So that's an important finding. And then we correlated the thickness of the dark band with the thickness of SRLM and dentate gyrus SM, and it was only significantly correlated with SRLM, but not with the thickness of dentate gyrus SM. So these results suggest that the dark band should be considered only as SRLM, so included in CA1, but not in the dentate gyrus label. So this is the kind of analysis we made for this project. And if you are more interested, you can read the paper, but we really hope that this observation will be used and will improve anatomical accuracy for future hippocampal subfield segmentation protocols. Yeah, you can stop me if you have questions, of course. Okay, so then I'm going to talk about this study that was recently published in Alzheimer's and Dementia, which is about the contribution of mixed pathology to medial temporal lobe atrophy in AD. From a neuropathological point of view, we know that AD is defined by the presence of amyloid plaques and nerve fibrillary tangles, and many studies showed that tau pathology, so nerve fibrillary tangles, were associated with MTL atrophy. So this was shown using ex vivo neuropathological measures of tau that was correlated with antemortem MRI, but also using in vivo CSF and in vivo PET. So this is pretty obvious that there is a strong relationship between tau pathology and atrophy, MTL atrophy. However, we know that there are additional pathologies in the context of AD, such as alpha-synuclein or TDP-43. So for example, about 50% of individuals with AD pathology also have TDP pathology. And importantly, these individuals with additional TDP-43 pathology will have an increased rate of hippocampal atrophy here in red as compared to people without this TDP pathology in blue. They also have increased likelihood of being cognitively impaired at death. And unfortunately, there's no in vivo biomarker for TDP pathology now. So we can detect amyloid, we can detect tau in vivo, but we cannot detect TDP-43. Moreover, the contribution of each pathology to MTL atrophy is not clear. So here we had two objectives. The first one was to examine the pattern of MTL atrophy associated with different co-pathologies. So basically the relationship between MTL atrophy and tau amyloid, TDP, and alpha-synuclein in the context of AD. And then we tried to discriminate patients with and without TDP pathology using MTL measures. So to do this, we used data from UPenn, consisting of 92 patients with AD pathology at autopsy. So for each patients, we had neuropathological examination, and we also had, so at autopsy, and we also had antemortem MRI with T1 images. So we could measure and estimate the pathology in the medial temporal lobe for these participants, these specimens. So we could derive semi-quantitative score for tau amyloid, TDP, and alpha-synuclein. So you can see here examples of mild pathologies and severe pathologies. And we used HST1. Well, this pipeline was designed to automatically segment medial temporal lobe subregions. So we used this on the T1 scans, and we could estimate the volume of the anterior hippocampus, the volume of the posterior hippocampus, and the thickness of the entorhinal cortex, BA35 and BA36, which are two subparts of the perirhinal cortex, and the parahippocampal cortex. And basically, we first correlated all these structural measures with all the neuropathological measures I've just described. So here are the results. You can see here each pathology that were correlated with each structures. And interestingly, tau pathology was associated with the volume of the posterior hippocampus, but it didn't reach bone-friendly correction. And TDP pathology was negatively associated with the volume of the anterior hippocampus, the volume of the posterior hippocampus, and the thickness of the entorhinal cortex. So this shows that there is an anterior to posterior gradient, kind of, with tau being more associated with the posterior part of the hippocampus and TDP being more associated with anterior part of the medial temporal lobe. So then we wanted to explore this, to know this association like more locally. So we used surface approach to look at the correlation between pathology and structure. So we only did this for tau and TDP because this is only with these pathologies that we had significant results, and we confirmed this pattern with tau being more associated with posterior part of the hippocampus, and TDP was more associated with anterior part of the medial temporal lobe. And then we compared patient with and without TDP pathology. So patient was considered as TDP positive when the score in the medial temporal lobe was above 1. And we could see that people with additional TDP pathology had smaller hippocampal volume, but also smaller volume, especially in the anterior hippocampus, and also smaller, like thinner entorhinal cortex. But we didn't find significant differences for the other regions. And interestingly, we calculated ROP to discriminate between these these two groups, and we saw pretty good AUC for the anterior hippocampus, which is nice, and which means that maybe the anterior part of the hippocampus could be good index to detect TDP-43 pathology in the context of AD. Yeah, so that's the conclusion, is that we found that tau was more associated, specifically associated with the posterior NTL, TDP with the anterior NTL in the context of AD. Once again, I think it's important. And the anterior NTL might be relevant to detect concomitant TDP pathology in AD patients, but of course we have to replicate this in independent population with a larger number of subjects, etc. And now I'll talk about this last project, and these results are super preliminary, but here we try to look at the association between functional connectivity and functional networks and atrophy propagation in the AD spectrum. So, so yeah. Yeah.
Sorry, Robin, there was a question here. We had a question about TDP-43 pathology. One, Wally asked if you could maybe just explain a little bit more about this. I just noticed the question.
Yeah, I'm not I'm not an expert of TDP pathology, but it's like tau, for example. It's a protein that will be hyperphosphorylated, and you'll have inclusion in neurons, and it will be associated with atrophy. And so it's not the main pathology in AD, but it's the main pathology in other form of dementia and other pathologies.
Okay. Jake?
I would have a question as well before maybe you continue to the next study. So to which extent do you think that, you're finding, so we know that Alzheimer's disease is so heterogeneous. So to which extent do you think that TDP-43 is really having an impact on cognition, since it's related to hippocampus? Or so to which extent do you think that this plays a role in the heterogeneity of the clinical expression of the disease? Did you check, or did you made any associate correlation, for example, with cognition or?
No. We didn't have any cognitive measures, and so we only included people based on the fact that they had AD pathology, but they didn't have. They were not all with AD dementia. Some of them had, FTD, like things like this. So we could, that would be interesting to check that.
Right, thanks.
But I think, because Joseph did it, we can probably find this information.
Absolutely.
Yeah.
Robin, I also had a question about this last study. So it was really cool. I was wondering, it's kind of a two-part question. The first part is, did you find that the individuals that had TDP-43 were older? Because I know that there's this idea of like late, which is like this age-related TDP that you often find as like a secondary diagnosis in Alzheimer's disease. So did you find that those individuals were older?
Yeah, they were older. That's right.
Okay, so I guess the follow-up question is that you've got, these forms of Alzheimer's disease that are older, often more associated with limbic pathology, like in the hippocampus and so forth. And you have, this kind of hippocampal sparing phenotype that's more associated with being younger. And so I'm just wondering if the effect you're finding is because older individuals tend to have, on average, more TDP and also maybe less hippocampal pathology to begin with, and maybe that's what's driving the association. I just ask because, in most cases, you would think that tau is appearing early in the anterior hippocampus. and kind of later on in the posterior hippocampus. So I'm just trying to find a reason that this association with the posterior anterior is so TDP focused in the anterior.
Right. Yeah, I don't really know. So we found differences in age, but it was not really dramatic. Like, and this group with TDP43 pathology had the same amount of tau pathology, so they were really comparable, but they just had additional TDP and they were slightly older. But I mean, we couldn't really see some huge differences between these two groups in terms of pathologies and things like this, other than TDP.
Got it. Okay, cool. Thank you.
Sorry, one last question. Sherry's asking, do you have any information about the type of TDP43 pathology that was present, type A, D?
Not really. I think they just measured. So I didn't do it myself because I'm not a neuropathologist, but I think they just measured tau pathology. Not a TDP, sorry. In general, not necessarily some specific type.
All right, thanks.
Should I continue?
Please.
Yeah. Okay. Yeah. So yeah, I was talking about networks and atrophy, and many studies showed relationship between brain networks and atrophy. And for example, in this paper, which is pretty famous, the authors showed the pattern of atrophy in different conditions. So in AD, but also in semantic dementia, for example. So these are the patterns of atrophy, and they took the regions with the most atrophied, well, this is the most atrophied regions, and they used these regions as seed to perform seed-based correlation analysis and to show networks. So all using resting state fMRI. So to show the regions that are functionally connected to these atrophy spots. And this is what you can see here. And it's very nice to see that there is a strong overlap between the networks and the pattern of atrophy in each condition. So it really suggests that there is a link between this specific pattern of atrophy in these different conditions and these associated networks. So many studies showed similar stuff. That there is a link between networks and atrophy. And interestingly, Ranganathan and Richard described in 2012 these two systems. These two systems on networks converged in the hippocampus. So there's the posterior medial system here in blue, which includes the posterior part of the hippocampal formation and the parahippocampal cortex, retrosplenial cortex, posterior cingulate, precuneus, angular, thalamus, etc. And there's the anterior temporal system, which includes the anterior part of the hippocampal formation, and the perirhinal cortex, and lateral betofrontal cortex, amygdala, and temporal cortex. So these two systems are pretty important because they are involved in different aspects of cognition. So for example, the posterior medial system is involved in episodic and autobiographical memory, scene perception, space and time, social cognition. And the anterior system is involved in recognition, associative memory, object perception, etc. We also know that these systems are targeted by AD pathology pretty early. Jack has studied these networks very recently. And we know that, for example, the posterior system is very affected in AD, while the anterior is more affected in semantic dementia. So these two systems have been studied quite a lot recently. And here we wanted to evaluate the structural covariance of different MTL subregions over time. And more precisely, we wanted to evaluate whether atrophy in the perirhinal cortex would covary with specific regions that are specifically involved in the anterior system, while the parahippocampal cortex would structurally covary with regions from the posterior system. So the idea here is that, you know, the atrophy of these two specific regions of the medial temporal lobe would spread and covary with a region from associated systems. So to test that, we used data from ADNI. So we used longitudinal data, and we included 117 cognitively normal amyloid negative individuals and 65 cognitively normal amyloid positive individuals, 148 and 1980 patients that were also amyloid positive. So like this, we covered all the AD continuum, and we could make sure that we had a lot of variability to study this structural covariance over time. So we used the T1 images to segment the medial temporal lobe subregions using HST1, and we could estimate the annualized change in thickness for BA35, which is a small part of the perirhinal cortex. So that would covary with the anterior system. This is our hypothesis. And we also could estimate the annualized change of the parahippocampal cortex here. We also applied ENS to calculate whole brain, to generate whole brain maps for longitudinal cortical thickness annualized change. And we basically used SPM12 to perform voxel-wise correlations between the annualized change of the medial temporal regions and all the voxels to look at the structural covariance between these anterior structures and the brain. We also used resting state fMRI. So these data were acquired at Penn. So we could use data from 68 cognitively normal elderly. And we just performed seed-based analysis to highlight these specific networks. So the anterior networks and the posterior networks. And for the anterior networks, we used as seed BA35, so part of the perirhinal cortex and the anterior part of the hippocampus. So we did this to make sure that we have enough signal in the seed, because there is often distortion in the perirhinal cortex. So we took bigger seed. And for same for the posterior network, we took the parahippocampal cortex, but also the posterior hippocampus as seed. And the idea was to compare the structural covariance and these networks to look at the overlap between the two. And these are results for the structural covariance. So we used uncorrected p-value of 0.001, but close to correction. And so we can see here that BA35 covaries with these regions in blue, which are mainly restricted to the medial temporal lobe. So when BA35 shrinks, these regions shrink as well. And the parahippocampal cortex structurally covariance with these regions in pink, which are more posterior and also including medial parietal regions. And we lowered the threshold quite a lot, and now we can see that BA35 covariance with almost all the left temporal region, and PHC covariance with all these pink areas, which are very, very posterior. So these are two different patterns of structural covariance for these two specific MTL regions. And then, oh, there is a question. And then we tried to highlight the specific networks. So, you know, we generated two maps, two connectivity maps, one for the anterior temporal network, one with the posterior. We compared them in SPM, voxel-wise analysis using paired t-test, just to show the specific anterior versus posterior systems. So we can see it here. The red is the anterior system, the green is the posterior system. It's in line with the description of Ritchie and Ranganathan. And then we just looked at the overlap between the structural covariance and these functional networks. And we can see here that BA35 structural covariance really overlap with this specific anterior network, and almost only this anterior network, while the parahippocampal cortex covariance, structural covariance, really overlaps with the posterior network. So it means that the structural covariance seems to really follows these specific networks, and atrophy seems to propagate within these specific networks. So that's it for now. We are still working on that, and we also want to look at the association between this propagation and tau and amyloid pathology in these networks, and also the links with cognition. That would be very interesting to investigate. And yeah, that's it. I just would like to thank all these great people from the lab from Penn. It was great to work with them. So yeah, that's it.
Thanks so much for a really nice and like really focused talk. It's really great. We have some questions. I know Gabe, you have one?
Yeah, I had a question about the very first topic you covered. You were discussing the kind of alignment, the problems with alignment with your slicing. So are there any complications due to malrotation of the hippocampus compared to the rest of the brain? And secondly, wouldn't some of these alignment problems go away if you did something like isotropic MRI instead of doing some kind of slab-based scan, which I think what you're doing?
Yeah, I'm sorry, can you repeat the first question, please?
So the first question was, for the very first part, you talked about problems with having the alignment of the scan wrong, so that you have these certain, so some people's hippocampi don't align up the same way, they don't give the same view that you wanted to for segmentation purposes. So is that due to like malrotation, or is that actually due to the scanner, just scanner alignment? And secondly, would this be a problem if you were to go with something like an isotropic scan instead, where alignment doesn't matter so much?
Yeah, that's true. Well, first, there's a strong variability in anatomy, so it's really hard to really capture the angulation that you really want to follow. And as you said, I think it's less a problem if you have isotropic images, because then you can rotate and have the exact angle you want to have. But The problem with this is that here we try to scan patients, and this sequences. Well, if you want to have really high resolution, then you need a lot of time, and you cannot do this with these patients. So, yeah, we made the choice to use anisotropic images, but having isotropic images would be really, really helpful for these problems, of course.
Do you have anything to follow up with, or we'll move on to Jake?
So one thing that was unclear about the alignment, did you prefer, when you're taking these slab scans, are you attempting to align with the ACPC or with the hippocampus? I can't remember which one was which for your preference. Like what do people typically do?
People typically align with the hippocampus and not the ACPC. And that's a problem because all the neuroanatomical literature is based on ACPC orientation. So there is a yeah.
But it's worth noting that design choice gets made to maximize coverage along the hippocampal main axis, so you can get more hippocampus voxels. Because if you make the choice not to have isotropic images, basically, right? So kind of these are all these trade-offs that they kind of come into play here. Jake, you had a question?
Yeah, thanks, Robin. That was a great talk. 3 really cool studies. I was wondering for your last talk, you know, with the original ATPM system, there's often this association with the default mode network in general with the posterior system. It's like posterior medial, so that's in the original formulation includes the anterior parts of the default mode network. But with your images and some of the other stuff that's been released recently, it seems like a lot of the kind of medial areas like cingulate and so forth aren't really involved so much with this posterior section. It seems like they almost kind of stop around the occipital and lateral parietal areas. So do you have any comment about you know what you think the DMN is, how it participates in this ATPM system and how your results kind of contribute?
So you're talking about these regions around there?
Yeah, I mean, I guess you have it there in the with the functional connectivity, where you do have a much lower kind of statistical correction. But yeah, I don't know. What's your thinking? I mean, because it says like the ventromedial prefrontal cortex and so forth in the posterior system as well in the original ATPM system.
Yeah, I don't really know. I think here I compared these two, I opposed these two connectivity maps to really show, try to show regions more connected with perirhinal cortex versus parahippocampal cortex. And you have these results, but if I remember correctly, when you don't do this and when you just look at the maps, so without comparing the two systems, you almost have a perfect DMN network.
So I guess you're kind of saying like if you look at the hippocampal area in general, you get DMN, but if you're kind of contrasting the anterior and posterior systems, it's not obvious how they participate.
Yeah, and even when you look at the connectivity map of the perirhinal and the parahippocampal separately, I mean, it's almost the DMN in both cases.
Cool, thank you.
So, yeah.
Are there any other questions from the audience? All right. If not, maybe we can all unmute ourselves and try to provide a bit of applause from home. So if you guys don't know,
Thank you.
a lot. That was a great talk. Thanks for taking the time to do it, given everything that's going on. I did not realize that you've only been able to go outside quasi-freely very recently. So thanks for doing this in light of all the other challenges you've got going on. Thanks very much, and thank you all for coming. And we'll see you all here next week, I hope. Take care. Bye, everybody.
Thank you. Bye-bye.